Good morning, dear two students. This is your teacher Neelam Elawat, and we'll continue our chapter, that is Human Health and Diseases. Okay, everyone, and this is our lecture number 6. I hope you are well preparing. Yes, your preparation is going on well, right? Okay, so let's continue our chapter, dear students. Today we'll be talking about another very most important disease, that is cancer. I hope you've heard this name. Yes, and some of the things you must be familiar also. So cancer is basically known as a malignant tumor. Okay, cancer is known as a malignant tumor, and this is a disease. This is basically a group of diseases where there is an abnormal growth of the cell is there. Okay, so in cancer, a particular cell or number of cells, there is abnormal growth. Okay, there is abnormal growth in those cells, and they then invade the other cells also, and then they spread to other parts of the body. Okay, suppose there is group of cells. Okay, these cells, they will multiply rapidly and form large number of cells, and this division is uncontrolled. They are multiplying on their own. So this is the uncontrolled growth of the cells, which later move to different parts of the body also. And cancer, dear students, it is considered one of the major cause of death all over the India. So it is a disease that causes causes a death all over the world. Okay, and because of its severity, right, because of this disease is very, very severe, its treatment and control requires extensive areas of research. Okay, so this requires extensive research in this particular area. And the cancer can be caused by the external factors also. See, we can divide that cancer can be caused by internal factors, or it can be caused by some external factors. Those factors, they are called as carcinogens. Okay, they could be any factor that can make the cells go out of control. Okay, so any factor could be a carcinogen. Now, if we talk about the worldwide trend, dear students, in humans, in women and men, we find that in case of women, the breast cancer, lung cancer, stomach, cervical, and colorectal cancers, they are most predominant. While in case of men, the esophagus, lung, stomach, liver, prostate, and colorectal, these cancers, they are mostly observed. And as per the data in 2007, the lung cancer causes death of 1.4 million people. Stomach, 8,066,000 people. Colon cancer causes, 6,77,000. Liver, 6,53,000 people. Breast cancer leads to a death of 5,48,000 individuals. Right, everyone? So this causes, if we look at the worldwide trend, so cancer results in a major death in the world scenario. Now, what are the causes of cancer? What are the factors that leads to the disease that is your cancer? Now, basically in this process, what happens? The normal cells, they become cancerous cells. Okay, normal cells, they become cancerous cells, and there could be 3 factors that could contribute to it. One is a physical factor, second is chemical factor, third is biological factor. On all these factors, they are called carcinogens. So talking about the physical factor, in physical factor we can say like ionizing radiations like X-ray, if a person gets exposed to X-rays, gamma rays or UV rays. So these can cause cancer, or it can make the normal cells to divide uncontrolled. Okay, so it can lead to the uncontrolled division of the normal cells. Second is your chemical agents like tobacco, if you smoke. Okay, then certain chemicals, certain tablets like which contain sodium azide, methyl ethane sulfonate. Okay, so the drugs, these chemicals, they again cause the lead to the uncontrolled growth or uncontrolled division of the cells, leading to the cancer. Third is your biological agents. There are some cancer causing viruses like oncogenic viruses, right? They are referred to as oncogenic viruses, and they have a gene which is called viral oncogene, which induces cancer. Then there are some cellular oncogenes which are also called as proto-oncogenes, right? When they get activated, when these genes they are active, they will act upon the cells and will make the division very, very fast. So the normal cells, they will divide at a very fast rate, leading to the cancer. Then what is a mechanism by which transforms a normal cell into a cancerous cell? Okay, we know that cell growth and its differentiation, it is highly controlled and regulated. Okay, it is highly controlled and regulated. Normal cells, they show a property of contact inhibition. Means, suppose this cell is there and these two cells there. If both of them they come in contact with each other, okay, right? If these cells they come in contact with each other, then this cell will act upon. Let's suppose this is A cell, this is B cell. So A cell will act upon the B cell and will regulate its division, while B cell will also act upon A cell and control its growth, right? So this is called contact inhibition. So normal cells, they have a property which is called contact inhibition. Now cells, the cancerous cells, they lose this property. ओके अगर ये दो सेल्स कैंसर हो जाते हैं तो ये कॉन्टैक्ट इनहिबिशन की प्रॉपर्टी को लूज कर देते हैं एंड देन दे डिवाइड uncontrollably, right? They divide uncontrollably and forms the tumors. Let's look at the picture. Let's look at the picture, everyone. Okay, let's look at the picture. Suppose in case of let's see, this is a normal cell. Okay, normal cells, they are there in the nutrient medium, right? Now slowly and gradually they'll multiply. Right, they were very far away from each other. There was large distance between the cells because the number of cells was less and medium was very large. So these cells, they will multiply, they will use the nutrients and they will multiply. Now see, all the cells, they have come in contact with each other, right? All the cells, they are touching each other. So each cell will exert an influence on the other cell and will stop its division. ठीक है, हर सेल दूसरे सेल के कांटेक्ट में है तो हर सेल दूसरे सेल को स्टॉप करेगा सेल डिवीजन से. ठीक है, क्लियर है? और ये सेल्स आगे डिवाइड नहीं करेंगे. क्लियर? If suppose there are cancer cells, suppose there are 3 cells, so see these cells will multiply and they will again form, they will again come in contact with each other. But here see, even they are in touch with each other, even then they are multiplying. देखिए ये सेल्स आपस के कांटेक्ट में है लेकिन उसके बावजूद दे विल मल्टीप्लाई बिकॉज इनमें जो कांटेक्ट इनहिबिशन की प्रॉपर्टी है वो लूज हो जाती है. दे हैव लॉस्ट द प्रॉपर्टी ऑफ कांटेक्ट इनहिबिशन एंड दस दीज सेल्स दे विल डिवाइड अनकंट्रोलेबली. ओके, एंड दिस विल रिजल्ट इन द फॉर्मेशन ऑफ ट्यूमर. दिस विल रिजल्ट इन द फॉर्मेशन ऑफ ट्यूमर. राइट एवरीवन? ओके. Let's compare a normal cell and a cancerous cell. Just look at the diagram, everyone. We're comparing both the cells. This is your normal cell, this is your cancerous cell. So normal cell, just look at the cytoplasm. See here, in both cases you see the cytoplasm in the nucleus. In case of a normal cell, the cytoplasm is large, while in case of dividing cell, cancerous cell, cytoplasm is small. Nucleus is single. Here there are multiple nuclei are there because it is undergoing a rapid division. Then single nucleolus is present. Here multiple large nucleoli are present. Chromatin fine, it is in the form of a fine details. Here it is in the form of a coarse chromatin is there. So we can easily compare the morphology or anatomy of a normal and a cancerous cells, and by analyzing the state of a cell we can tell whether it is a normal cell or whether it is a cancerous cell. तो अगर हम दो cells को compare करते हैं और if we know the characteristic of a normal and a cancerous cell, so we can identify that this particular cell is a normal or a cancerous cell. Okay, now let's look up the second diagram. Just look at the second diagram, everyone. These are normal cells, so they will multiply continuously. और जैसे ही वो एक दूसरे के कांटेक्ट में आते हैं वो मल्टीप्लाई करना बंद कर देते हैं. बट इन केस of cancerous cells, they will multiply continuously, right? They will multiply continuously. Okay, so these cells, they will multiply you can say uncontrollably or continuously, and they will not follow the contact inhibition, and this will result in the formation of a tumor. Okay, this will result in the formation of tumor. Yes, everyone? Okay, so coming to the types of tumor. Basically, dear students, there are two types of tumor. One is your benign tumor. Okay, one is your benign tumor. Second is your malignant tumor. Okay, so first is benign tumor and second is malignant tumor. Now, benign tumor mostly, here normally the cells, they are confined to the original location. Means they do not move from one part to the another part. वो एक ही particular location पे रहते हैं, spread नहीं करते हैं. And these cause a little damage. They do not cause the major damage. They only cause the little damage. Malignant tumors, they are very, very important because they are the ones which will be affecting our body in a very large amount. So these cells, they grow rapidly. They invade and damage the surrounding tissues. Okay, तो ये एक जगह से दूसरी जगह मूव करते हैं, सराउंडिंग टिश्यू को डैमेज करते हैं, and they divide actively and grow. And they also starve the normal cells. ये इतना ज़्यादा न्यूट्रिएंट मीडियम को यूटिलाइज करते हैं because they are continuously dividing that the normal cells, they do not get the food and they are starved of the nutrition. And these cancerous cells, they escape from the site of origin. ये एक पर्टिकुलर प्लेस है जहाँ ये फॉर्म हो रहा है, वहाँ से दे विल मूव टू अदर प्लेस थ्रू ब्लड. सो एक प्लेस से दूसरे प्लेस में पहुँचते हैं थ्रू ब्लड के थ्रू, एंड देन हियर ऑल्सो दे स्टार्ट मेकिंग द कैंसरस सेल्स. एंड दिस प्रोसेस ऑफ मूवमेंट ऑफ मेलिग्नेंट ट्यूमर्स फ्रॉम वन प्लेस टू अनदर एंड देन स्प्रेडिंग द डिजीज देयर इज कॉल्ड द मेटास्टेसिस. ओके, दिस इज कॉल्ड द मेटास्टेसिस. राइट एवरीवन? Now, how can we detect cancer? How the cancers, they are detected chemically? So, first technique is biopsy and the histopathological study. Now, इसमें हम क्या करते हैं, हम टिश्यू या ब्लड का सैंपल लेते हैं, ठीक है, और उनको टेस्ट करवाते हैं कि उनमें जो रेट ऑफ मल्टीप्लिकेशन है वो ज़्यादा तो नहीं है. दे डोंट हैव अ, एज आई टोल्ड यू, वी कैन कंपेयर द नॉर्मल सेल एंड द कैंसर सेल. तो हम सेल्स को कंपेयर कराते हैं कि कहीं किसी पर्टिकुलर पोर्शन में कोई सेल एबनॉर्मली तो डिवाइड नहीं कर रहा है, ठीक है. तो हम एक पीस लेते हैं जहाँ से भी जिस भी पर्टिकुलर पोर्शन में डिटेक्शन चाहिए हमें, हम उस पर्टिकुलर सस्पेक्टेड टिश्यू से एक पीस लेंगे, विल स्टेन द सेक्शंस एंड एग्जामिन अंडर द माइक्रोस्कोप and will distinguish whether it is a normal cell or a cancerous cell. Second is radiography like X-rays, computerized tomography. Now in this case, the cancerous cells, it can be used to detect the cancerous cells for the internal organs. Okay, हमारे internal organs में कोई cancer spread तो नहीं हो रहा है. Then computed tomography uses X-rays. इस process में जो है X-rays utilize होते हैं और उससे एक 3-dimensional image generate होती है हमारे internal organs की, और we can easily compare that 3-dimensional image with the normal image, and if there is any discrepancy we can easily identify that. Then third is MRI, that is magnetic resonance imaging, right? This is called magnetic resonance imaging. Now this basically uses strong magnetic fields and ionizing radiations so that it can detect the pathological and physiological changes. Okay, so it can detect the physiological and pathological changes that are occurring in the body. The second way is antibodies against a cancer specific antigen. See, we all know, dear students, that whenever certain abnormal things happen in the body, whenever certain foreign material enters the body, then our cells, our immune system, it synthesizes antibodies. Okay, so we can detect those antibodies if they are being made in our body. Okay, हम ये चैनलाइज कर सकते हैं, हम ये डिटेक्ट कर सकते हैं कि क्या हमारी बॉडी में जो है कैंसर के जीन्स के खिलाफ कुछ एंटीबॉडीज़ बन रही है व्हेन वी एक्सपोज देम. ओके, क्लियर? सो अगर एंटीबॉडीज़ प्रेजेंट हैं देन इन सम पार्ट ऑफ आवर बॉडी देयर आर सम कैंसर सेल्स. right? Then treatment of cancer basically involves 3 therapies. One is your surgery, second is radiotherapy, and third is chemotherapy. Surgery में we'll remove the cancer using the surgical method. Here we will kill the cancer cells by using radiations, and in chemotherapy the cancer cells, they will be destroyed by using drugs. Let's come to the surgery. This is your surgery method. इसमें क्या करते हैं, हम ट्यूमर सेल्स को रिमूव कर देते हैं. सपोज दिस इज योर ट्यूमर सेल्स, सो दिस विल बी रिमूव फ्रॉम द बॉडी. रेडिएशन थेरेपी में हम इस तरीके से पेशेंट को जो है इरेडिएट करते हैं उस पर्टिकुलर पोर्शन पे जहाँ पर उसकी बॉडी में कैंसर सेल्स हैं, जिससे कि नॉर्मल सेल्स को डेड ना हो, बल्कि रेडियोथेरेपी इन्वॉल्व हो जाए और कैंसर सेल्स जो है वो डेथ हो जाए उनकी. सेकेंडली कीमोथेरेपी, इसमें हम पेशेंट को जो है सिरेंजेस के थ्रू थोड़े कुछ ड्रग्स इंड्यूस करते हैं. क्लियर? सो यहाँ पर हम कैंसर सेल्स को किल करते हैं यूजिंग अ स्पेसिफिक केमिकल. लेकिन इसके कुछ साइड इफ़ेक्ट्स होते हैं, लाइक हेयर लॉस, एनीमिया. ठीक है, आप में ब्लड की कमी हो सकती है, आपका हेयर लॉस हो सकता है. सो वी शुड यूज़ द डाइट that a patient is using, that should be very quality one. Quality diet should be used in case if you are undergoing a chemotherapy. Then is your immunotherapy. इसमें हम क्या करते हैं, हम अल्फ़ा इंटरफेरॉन्स को एक्टिवेट करते हैं हमारे इम्यून सिस्टम में. ओके, जो हमारे कैंसर सेल्स को डिस्ट्रॉय कर देते हैं. ठीक है, तो हम अपने अल्फ़ा जो अगर किसी पर्टिकुलर सेल में कोई सपोज़ ये सेल है आपका, तो इसके ऊपर अगर कोई वायरस से इन्फेक्टेड है तो ये एक सर्टन केमिकल्स प्रोड्यूस करता है जिसको हम कहते हैं अल्फ़ा इंटरफेरेंस. तो अल्फ़ा इंटरफेरेंस जो है ये आपके इम्यून सिस्टम को एक्टिवेट कर देते हैं, एंड देन योर इम्यून सिस्टम विल ट्राई टू किल दोज सेल्स. ओके, सो मोस्ट ऑफ योर यू कैन से cancers they are treated by using a combination of chemotherapy and radiotherapy. सो कॉम्बिनेशन यूज़ किया जाता है, एक पर्टिकुलर you can say therapy को ना यूज़ करके combination of these therapies used, because it is suggested that doctors have suggested it that it is better that we use a combination of these therapies. Coming to your homework, dear students, first of all you will make notes of the topic and then you will read the page numbers 157 to 158. Okay everyone, you will read the page numbers 157 to 158, and if there is any doubt you will send your queries to me, right everyone? Okay, so that's all for today. Bye everyone.